Hello everyone, I'm Darshana. So in this video, I thought of again discussing a topic related to the COVID-19 situation and I mean there has been a lot of talk regarding the vaccine. So I'm living in Sri Lanka where the Oxford-Astrazeneca vaccine is the one that is approved, and I think that vaccine is approved in the US as well as the UK. So now I came across this uh, discussion regarding the Oxford-AstraZeneca vaccine, and some people were arguing that it can, I mean, whether it alters your DNA, whether it can modify your DNA, which I thought was kind of a funny question because after all, it's a vaccine. So, what does it have to do with the DNA at all? But so I'm not an expert in this domain, but when I read some of the public domain material, I felt that it's not such a ridiculous question to ask in the first place. I mean, that's what my impression was, and also I don't think it has been definitely answered in a definite manner that is proved using some kind of an experiment method that it does not do that. So, so I'm going to share some of the public domain information that I found, and you can think about it and you can share your thoughts and comments. I think it's a very important and interesting question because it seems vaccines, COVID-19 vaccines, as well as COVID-19 will be part of our lives for a foreseeable future. So let me share that content with you. Now, so this is actually a UK government from a UK government website, the Medicines and Healthcare Products Regulatory Agencies uh, publication, right? That's the apparently that's the regulatory body in the UK to regulate vaccines and healthcare products. So there they have a kind of a memo from January 28th, right? Information for healthcare professionals on COVID-19 vaccine AstraZeneca, which is a vaccine that is approved in the UK, US, and some other countries. Yeah, it's kind of a regulation for healthcare professionals with some information, right? Name of the medicinal product, it's so this is the official name, Chad OX1S recombinant. So this word recombinant is important. I'll come back to that in a moment, and then it says qualitative and quantitative composition, right? So one dose 0.5 milliliter contains COVID-19 vaccine, again that name, and this many viral particles, right? So it's effectively it's another virus, right? It's another virus, and it is actually recombinant replicant deficient chimpanzee adenovirus vector encoding SARS-CoV-2 spike glycoprotein producing genetically modified human embryonic kidney HEK293 cells, right? So there's a lot of information there. One thing is this recombinant DNA. So apparently recombinant DNA, I mean this is just from Wikipedia, but I think this is kind of standard knowledge. So molecules are DNA molecules formed by laboratory methods of genetic recombination, such as molecular cloning, that brings together genetic material from multiple sources, creating sequences that would not otherwise be found in the genome. So it's basically a recombinant DNA is some kind of a new organism that, or you can create a new organism using recombinant DNA. So basically genetic engineering, right? And it's from this chimpanzee virus that is found in chimpanzees, and so I found that this nice diagram in New York Times, and they have actually cited it from I think some credible sources, right? The Nature magazine and all that. So they graphically show this. So you have the coronavirus and the spike proteins. I think spike proteins are the ones that kind of enable the virus to get attached to the host cell, right? Human or any other host cell. And so the adenovirus is apparently a virus that is found in chimpanzees, right? So it infects them, but it does not infect humans. And the scientists have, AstraZeneca scientists have modified the adenovirus so that the COVID-19 spike protein is combined with the adenovirus, right? So then it can infect the humans. And so that's the idea. Now, so this is kind of, I think this the technical term for this is gain of function research, where you take some naturally occurring virus that does not affect another species, maybe humans, and then you can, genetically modify it to infect humans. So it's kind of interesting because I think some scientists had raised the concern whether even the SARS-CoV-2 virus is developed in a lab, right? So it wasn't a naturally mutated virus. It's something that developed in a lab. So I think they did some genetic analysis to do that, but only a handful of scientists, I guess. Now, regardless of whether that is true or not, I think certainly the technology is there, and this shows that it is theoretically, or the technical capability is there to do something like that, right? And so that is there, right? And this is called replication deficient, right? So I think what that means is, so there's this nice resource in YouTube called channel called MedCram, which actually explains these ideas in a very simple manner, and it's done by Professor Roger Seheult. I think I can't really pronounce his name, and let me bring up his profile. He's from University of California, Riverside School of Medicine. So he's an associate professor there. So he actually explains this process very nicely, how the AstraZeneca vaccine works in with nice diagrams, right? So and he explains that this is what this term replicant deficient means. Basically, it means that the adenovirus has been genetically modified so that it cannot replicate itself, so that it doesn't become a new disease that affect humans. When you inject that virus, it's not supposed to create a new disease, right? So because of that, because it can then mutate and create some new disease. Right. So, and it says this product contains genetically modified organisms. So it's basically GMO product. Now, some people have some reservations regarding GMO products, particularly vegetables and things like that, or meat, because there are some concerns whether they are kind of they would create some chronic disease, right? Anyway, this one is also a genetically modified organism, right? It contains genetically modified organisms like a live virus. And the other thing is it's produced, so it's kind of, you need some culture for the virus to survive, right? In the shell storage and all that. So it is kind of cultured in genetically modified human embryonic kidney HEK 293 cells. So this HEK 293 cells there's an interesting backstory to this. So basically, HEK refers to human embryonic kidney 293 cells, which is uh, uh, so basically it's taken from aborted female fetus back in 1973, right? Although this abortion happened in 1973, and so that tissue has been cultured, and that is the medium, right? That is the medium in which this adenovirus survives, right? So that too apparently gets injected, right? Part of that, because that's the culture. And so yeah because that's just part of the vaccine. Now, So those are some background information, and it says that there's something that I found interesting here. So it says that elderly population, in the same article, the efficacy and safety data are currently limited on individuals greater than 65 years of age. No dose adjustment is required. So it doesn't say that you shouldn't take it if you are greater than 65, even though the data for efficacy and safety is limited, which is an interesting thing, I thought. And I think because of this, some European countries have not recommended this for all the people. Again, I think it's kind of unfortunate because that is the age group that is supposed to be most affected by COVID, and at the same time, it says that the safety and efficacy data are limited, which is an interesting thing. The other thing is, so it's a uh, so it's supposed to be given, according to this article, it's supposed to be given in two separate doses, which are like 4 to 12 weeks apart, right? Right. Now I think in my previous video, I think I mentioned about this YouTube alternative called Rumble, which is kind of I mean, it has less censorship because these kind of topics are very controversial, you know, and so I found this particular video in Rumble, which talks about this. I mean, it contains this adenovirus and aborted fetal like cells cultured from an aborted female fetus and so on. Now, so this is an interview with a doctor, person called Dr. Brian Hooker, and so this is his CV, and he apparently has a lot of experience in biotechnology and biochemical engineering, and he has apparently published around 65 papers in this domain, and also he has 5 US patents. So I think he has, I mean, uh, I mean, quite broad knowledge in this domain, it seems. But he's a controversial figure, and I'll come to that later on. Um, he's no stranger to controversy. But anyway, he raises some issues with this information, about the potential risk of the AstraZeneca vaccine. So, so one of the things that he raises is this, um, whether these genetically modified organism can alter the DNA. I think I'll come to that in a moment. So because everything that we discuss related to Brian Hooker is related to the idea that whether the vaccine itself can alter the human genome. Now, I'll bring up um, another article from this website called Very Well Health. So it is actually, uh, I think it's a quite rich, credible resource because it is it is a resource that has like with a review board of around 100 specialist like consultants, and it is actually affiliated to the Cleveland Clinic in the US, right? So which is one of the top hospitals in the US, and has received multiple awards uh, re- regarding disseminating medical information. So it looks like I think it's a pretty credible resource. Um, and so there's this nice article about what's the difference between a DNA and a RNA vaccine. So we know that there are three types of vaccines that are approved in different countries. Two the Pfizer vaccine and Moderna vaccine, which are called RNA vaccines, so mRNA vaccines, and uh, Oxford-Astrazeneca vaccine is called a DNA vaccine, right? Now, the difference between the two um, is uh, is explained here by again Professor Maria Gennaro. So I think I'll just so this is her profile. So she is a professor in medicine and epidemiology at Rutgers, New Jersey Medical School, and so yeah, maybe I'll just jump back to sorry for like jumping back and forth. So I'll jump back to this YouTube channel. So by Professor Roger Seheult, and there he explains of AstraZeneca. So he calls it AstraZeneca DNA COVID-19 vaccine, right? Because it's essentially a DNA vaccine. And so the operation, apparently how this happens is yeah, I'll come to this. I'll use this New York Times article. So this adenovirus, so it has the spike protein. It is genetically modified to have the spike protein of SARS-CoV-2, right? So it's kind of a hybrid now, and so because of that, it can enter the human cell. It can effectively infect the human cells, and then these messenger RNA that contains the information about the spike protein, right, gets injected to the nucleus, right, cell nucleus. So it has to like penetrate the cell nucleus, and from the cell nucleus, then the messenger RNA is kind of emitted. I'm not sure these are the proper technical terms, but it's just a layman's understanding. I mean, I'm a layman when it comes to this domain. So then this messenger RNA is emitted through the cell nucleus, right, and is will create us the human self to have the spike protein, which is similar to the SARS-CoV-2 spike protein. So then our immune system will identify this like a, I mean, this it will identify the cell as some kind of a alien entity, right, attacking the body, and it will develop antibodies and right destroy the cell, right. So that's the idea. So it's essentially it's different from the traditional vaccines, because I think I'll come to that in a moment. So that's the idea, right? So it's because I think in traditional vaccines, what happens is you give a weakened form of the same virus or the bacteria, and then the immune system directly develops the antibodies. But here it's kind of done in a roundabout manner. You first, in the DNA vaccines, you uh, inject some genetic information to the cell nucleus, right, where the DNA resides. So our genetic information is there in the DNA, in the cell nucleus, right? And so that's where the genetic information is there, and the messenger RNA is injected to that, and through that you kind of indirectly change the cell to behave like a viral cell, or kind of a infected cell, uh, and then you expect the antibodies will get developed, so that when the antibodies will be there when the actual wild virus, you encounter the wild virus in the environment, you will have the immunity, right. So this is the idea. And in contrast, if you look at the how the Pfizer-BioNTech vaccine works, which is RNA vaccine, so it's different. So here the spike proteins, they get injected to the messenger RNA that is capable of producing the spike protein, is directly injected to the body through some medium, and then that gets into the cells, and then using the cell machinery, it supposed to produce the same similar spike protein, right, so that the immune system will develop antibodies for that. The key difference is that there's no adenovirus like virus sort of infiltrating the cell nucleus. So the mRNA just uses the cell machinery, and produces this protein, spike protein, that will uh, appear in the human cell, right. So right now, so with that background, we will go back to this um, article. So it's verywellhealth.com article, and he actually so Professor Maria Gennaro talks about the difference between these two vaccines, right? And there are some important notes here that says that a DNA or RNA vaccine has the same goal as traditional vaccines, but they work slightly differently. Instead of injecting a weakened form of a virus or bacteria into the body, DNA and RNA vaccines use part of the virus's own genes to simulate an immune response, right? So in other words, they carry the genetic instructions to the host cells to make antigens, hosties. In this case, it's a human, right? Right. So Professor Maria Gennaro says that so far there has been no mass production of vaccines based on DNA or RNA, right? Maria Gennaro, MD, Professor of Medicine. So this is kind of new, so that's her thought. Right. And it highlights the differences between the two. Pros and cons of DNA and RNA vaccines. So DNA and RNA vaccines are yeah, I'll go to the risk section, right? Because the other things are about the cost and all that. But with any health advancement that comes potential risk, right? Gennaro says that with a DNA vaccine, there's always a risk that it can cause permanent change to the cell's natural DNA sequence, right? Because this, I guess, of this operation where the adenovirus it has to sort of infiltrate the nucleus to so the cell is supposed to produce the mRNA through the cell, right? So you have to infiltrate it, otherwise it won't work. Usually there are ways in which DNA vaccines are made that try to minimize this risk, but it's a potential risk. Instead, if you inject mRNA, it cannot get integrated into the genetic material of a cell. It is also ready to be translated into protein, right? So like I said, with the mRNA, the mRNA does not infiltrate the nucleus, which is where the genetic information is. Because no DNA vaccine is currently approved for human use, there's still much to learn about the effectiveness. With 2 mRNA vaccines in phase 3 trials and 1 approved for emergency use, they are much closer to full approval and licensing by the FDA. So this article was published in December 23rd last year. So even at that time, I mean, essentially all 3 vaccines, the 2 mRNA vaccines by Pfizer and Moderna, and the DNA vaccine by Oxford-Astrazeneca, all these are like the first of its kind, right? Or its kinds, because that's a novel technology. So that's something that I want to highlight. Now I want to jump back to Dr. Roger Seheult, this MedCram channel, and where actually the title says coronavirus update, AstraZeneca DNA COVID-19 vaccine, right? So basically, this is an Oxford-Astrazeneca vaccine is a DNA vaccine, right? And also now this concern raised by Professor Maria Gennaro. But yeah, I'll let me jump to another source. So this is by the this is article by in the CDC website, right? Centers of Disease Control and Prevention, that is US's regulatory body for vaccines, and it's about the mRNA vaccines. So that is Pfizer and Moderna vaccines, which are the most common vaccines in the US as I understand. And so here actually it says give some facts about COVID-19 mRNA vaccines, which is trying to address some of the concerns of people like this. First one is they cannot give someone COVID-19, obviously, because it's using this RNA sequence, right? And unlike the Oxford vaccine, it doesn't use this adenovirus. The second point is they do not affect or interact with our DNA in any way. So that's the important part for our discussion. and under that it says mRNA never enters the nucleus of the cell. Under that it says mRNA never enters the nucleus of the cell, which is where our DNA genetic material is kept, right? So that's the important point. And the second point is the cell breaks down and gets rid of the mRNA soon after it is finished using the instructions, which is an important note. I'll come to that later, but this is the one that I want to. This is the one that is relevant for our discussion. So the basic argument that they give here is that the reason why mRNA vaccines like Pfizer or Moderna vaccines, they don't affect our DNA, the justification for that is that mRNA never enters the nucleus of the cell, right? Which is how it is supposed to work, right? So it uses the cell machinery, but it doesn't go into the nucleus. But now with that argument, you can, someone can raise the question. So how about the DNA vaccines? Right? Because in the DNA vaccines, it is designed to the adenovirus is supposed to put some genetic information to the nucleus, right? Otherwise, the mRNA vaccine will not be produced. mRNA would not be like emitted. I'm not sure whether the technical term for that, but I mean you have to go through the cell, right? So you have to infiltrate the nucleus. So then you can question whether this, if you apply the same argument to DNA vaccines, whether this will actually affect the nucleus, right? Whether it will affect the nucleus or whether it I mean, as we understand, based on this public information, definitely the new cell nucleus is breached, but the problem question is whether it will alter the or interact with the genetic information in the cell, right? So so that's the interesting. So that's the interesting source, and that's the interesting claim that I found in the CDC website, which is relevant to our discussion about Oxford-Astrazeneca vaccines, right? Now, in this YouTube video by Professor Roger, he actually very briefly addresses this issue, saying that AstraZeneca has claimed that the DNA does not get altered by their DNA vaccine, right? Now, I'm not sure whether it has been published, whether such experiment has been done or whether it has been published, and I'd be happy to know whether if you can find any research done on that to prove that the DNA sequence of the recipient is not altered from the vaccine. I would be interested to know that, because then then this is a done deal, right? So I think it should be a relatively easy thing to test. I'm not an expert in this field, but I think DNA sequencing matching has been an area that has been a fairly developed area over few decades now, I guess, at least couple of decades. So I think it should be fairly easily testable hypothesis whether it actually alters the DNA. So I think it should be tested because it's a interesting problem. But yeah, so that could settle the problem. So, but I haven't seen so far any published research on that. Right. Another point that I want to highlight is that some people are reporting that they had adverse reactions like fever, diarrhea, and nausea, and vomiting after taking the vaccine in the social media. So the general understanding is that it's normal, that it's the antibodies kicking in, and it is how you know that vaccines work, right? But as a layman, I'm just wondering whether some of those reactions may be due to this process, right? When the adenovirus enters the cell and even infiltrates the cell nucleus to inject this mRNA. I suppose the body would kind of react to that, because that's not how cells would typically try to protect the DNA, because the DNA contains genetic information, and then only the antibody reaction is supposed to happen after that, right? After the mRNAs display these spike proteins, and then only the antibodies should kick in. So maybe, I mean, as with the kind of the official explanation, the creation of antibodies and that reaction might also have some make the person feel some reactions. But I'm just wondering whether at least some of those reactions may be due to this whole process of injecting the mRNA um, into the cell nucleus, right? Because that, I suppose, the body would react in some way. I'm not really sure about that, but uh, uh, whether at least some of the reactions are due to that whole process, right? Or uh, some of the reactions may be, yeah, maybe bodies, how the body responds, right, to this. Because for the body, I think it might seem like an attack. Some alien agent is trying to, inject um, some DNA, some genetic information to the cell nucleus. So I suppose the body will have some kind of reaction to that, so reactionary or protective mechanism. That, and some of these symptoms, whether some of these symptoms could be a manifestation of that, right? So that's something to explore further, I think. Yeah, so that's basically what I want to talk about um, regarding this topic. uh, I will talk about some other things later on. So basically, from this public domain information, my current understanding is that uh, so the idea that the Oxford-AstraZeneca vaccine, or any rDNA vaccine, any recombinant DNA vaccine, the idea that they can alter the DNA, or they might alter the DNA of the host, of the recipient. I don't think it's a dumb question, or it's a ridiculous question or a crazy idea. I think my feeling is that it's definitely plausible, and I think it's something that can be easily validated, right? So if it doesn't affect the DNA, then I think everybody would be happy, because now, I mean, there's mass scale distribution and prescription of this vaccine. So already many people have received it. So, but I think we ought to test it. That's my opinion, and I think it should be testable, right? Now the other question is, so what if? I mean, okay, what if uh, uh, it actually alters your DNA, right? The DNA, it can alter your DNA. So what's the big deal? Because that's also a valid question. I mean, is it a problem? So that's also a valid question. Is it a problem, right? So now I'll come back to this interview by David Hooker. So he actually raises. Actually, he is raising the issue with the using this HCK cells, human tissue, uh, like from this aborted female fetus from back in 1973, because he says that there's a possibility that even this human DNA can get attached to the DNA of the host human cell, because the nucleus is now breached, right, from this adenovirus, because DNA is very malleable. So I'm not an expert in this area, so I can't really comment on that. But apparently he has worked a lot with genetic engineering, so I think there may be some point to that. So um, so this from this female fetus, yeah, whether that DNA can get kind of attached to existing DNA. So that's also interesting problem. The other thing is, so he is actually raising some concerns uh, regarding the Oxford-AstraZeneca vaccine, supposing that it alters the DNA, right? So one thing that he points out is, uh, whether that can cause abnormal cells, and the most common fear is that whether it can cause cancer. And whenever you alter genetic information in a cell, whether it can create abnormal cells, and it can make them replicate and cause cancer, right? Or any other chronic disease. That's number one. Number two is the so that's kind of a general argument regarding the efficacy of vaccines. I think not just this vaccine, because what he says is when you have one population that is vaccinated, because when you roll out a vaccine, it takes some time. So one population will be kind of immune to the existing virus, COVID virus, SARS-CoV virus, and then so that virus will evolve in that population, and eventually it will get more virulent, right? More deadly, so to speak, because you are kind of, it is evolving in different populations, and then it can jump to the other population and so on. So I think that's kind of a more generic argument regarding whether viruses can actually achieve herd immunity, because that is kind of I think that argument is kind of supported by what is happening with flu vaccines. So every year in the Western countries, in cold countries where you have a cold winter, give flu vaccines, but every year you have to give a new vaccine because apparently the virus also evolves, right? So his basic argument is when you do this rollout of vaccine, usually the virus will mutate and become even more virulent, right? More vicious, so to speak. So that's kind of a general argument regarding the whole idea of vaccines. The third point that he's raising is this idea of antibody-dependent enhancement. So I think I talked about that in the previous video, where because here actually kind of the human cell is altered to have these spike proteins similar to the SARS-CoV-2 spike proteins. So the idea is if you encounter the wild virus, then immune system will respond to that. At the same time, immune system will sort of recognize all these human cells as potential threats, and basically attack the body itself. It will be like an autoimmune disorder, right? And which can create make people go to a very serious condition in a fairly short time, right? So that's, I think that I explained that argument in the previous video. You can refer that. The final one is an ethical one. I mean, here because you are using the aborted female fetus tissue culture. I mean, that is a medium effectively. So some people might be offended by that. I mean, particularly people who are opposed to abortion, like the evangelists and so on, and they might not know that it actually contains this these kind of tissue, right? So most of them may have even got vaccinated, right? Because this information is not that it's there, but you have to look for it, right? It's not something that you find in the media, for example. Right. So those are, I think, all those are valid points. I think so. Three of them are like specific to the DNA vaccine. One of them is like a general point against this whole idea of vaccination to achieve herd immunity, whether it can make a virus more virulent, right? So I mentioned that there's some background to this person. I'll come to that later on. So basically, that's what I want to say. And there's another thing that I want to highlight here. In the CDC article regarding mRNA, it says that the cell breaks down and gets rid of the mRNA soon after it is finished using the infrastructure. So this whole idea is mRNA is very kind of vulnerable, because that's why you have to store it in a very cold temperature, and it says that it breaks down quickly after entering the body. So this is not regarding the Oxford vaccine, this is regarding Pfizer, Moderna vaccines. Now I found another video from this controversial resource, Humble, Yeah, there's another controversial person, a neuroscientist, Christopher Shaw, from professor from University of British Columbia. So he is also not no stranger to controversy, and so he's actually still there. I mean, at UBC as a professor in neuroscience, right? And so he is raising this issue uh, regarding this claim, not specifically regarding this claim, but there's this general understanding that mRNA will quickly kind of break down, so that does not go into other organs, and that does not cause any harm, right? So he's citing this paper. Hmm, yeah, preclinical and clinical demonstration of immunogenicity by mRNA vaccines against influenza virus. So this is a paper published in 2017, and it's actually done by sponsored by Moderna, right? So it's a paper published by Moderna employees. So they should be very familiar with it, and it's regarding mRNA vaccine, but some other vaccine, right, against some other virus, influenza viruses, right? So here actually in table 2, so this is an animal study based on using mice, right? Table 2, it shows that how the mRNA kind of residue is found in different organs of these animals, right? Brain and all this. So basically it's there everywhere, right? Uh, and it's small, they are in very small quantities, but the point is we don't know how. So what Professor Chris Shaw says is we don't know how much is safe, and it is not supposed to be there, right? By the current understanding of the mRNA, it should break down quickly in the muscles. So it's there in the brain. So he says that whether it can cause inflammation in the brain, because I mean it's not supposed to break the blood-brain barrier, right? And one of the ways that the brain reacts to any alien material coming is through inflammation, right? So that is another interesting thing I found regarding this claim, right? Because if it breaks down so quickly, how come it ends up in different organs? Now, I'm not sure whether a similar study has been done regarding the current COVID vaccines. I think it should be possible, at least using the animal studies. Because with humans it's difficult to, because you have to kill the subject here to see how the organs are doing. Yeah, that's another thing that I want to highlight. So those are the two things that I want to highlight, and do share your thoughts. And I'll just add some some additional material, full disclosure. So um, um, to highlight that some of the, I mean, give some background information about some of the scholars and some of the academics that I spoke about. So Professor Roger Seheult, I don't think he's a controversial figure. He's basically a mainstream, professor. There's no controversy around him, and so is Professor Maria. Right? So now, when it comes to Brian Hooker, that's a different story, because I will give that background, so you can decide for yourself whether these people have kind of a vested interest against vaccines, or biased against vaccines, and but regardless of their history, I think what the points that they claim in these two interviews, I mean, as a layman, to me that sounds like valid points, right? But anyway, I will give you that background as well. So, but Brian Hooker, he was quite came into the spotlight. I mean, he was a researcher, but people started to talk about him because of this documentary called Vax, right? So this is actually, so what happened was, there's this scientist called William Thompson, who works at the CDC, US as regulatory body, and he was an author. He was an author of this journal article, which was published in Pediatrics, Age at First Measles. So the idea is that there were a lot of court cases in the US from parents arguing that the MMR vaccine, measles-mumps-rubella vaccine, has caused autism in children, right? So because of now, usually these regulatory bodies operate in a very reactive fashion. They want they would give the approval based on the studies done by the pharmaceutical companies, but only if there's a legal issue, some public commotion, they would do their own research, right? So here CDC opted to do their own research to settle this issue whether actually MMR causes vaccines, autism. So William Thompson is one of the authors there, and the paper showed that there's no correlation between the MMR vaccine and autism, right? So things got back to normal. So this was published in 2004, and I think several years back, William Thompson contacted. So he's still at CDC, and he's also a very accomplished researcher, it seems, right? A lot of citations. He contacted Brian Hooker. Maybe they are colleagues. Maybe they know each other because they are in the same field, right? And shared some information saying that very objectionable information saying that there was actually a correlation with MMR vaccine and autism, but they kind of fudged the data. They omitted some results to make it look like there's no correlation, right? So now there were some telephone conversations. Apparently, Brian Hooker, recorded these conversations, and so at one point there was a fallout, and and then Brian Hooker, as a whistleblower. So in the US, apparently there's a whistleblower law where you can, if you feel that something fishy is happening in your organization, private or public organization, you can get whistleblower protection. So he basically sought legal assistance, right? So William Thompson, through his law firm, so I'll share this article, issued a statement saying that this happened. I was part of it, and I talked to and apparently he shared the original data, which he claims to be the original data, with Brian Hooker, and so this legal statement is there to protect himself, I guess, and Brian Hooker then published a different article in another journal, which was actually retracted. So this is the open access version, but the other version is retracted, saying that yes, there is a correlation based on the data that William Cooper gave him, right? So this was actually covered in some of the media and in the. So yeah, there's an article published in Forbes back in 2015 regarding this whole saga, and also these documents leaked by William Cooper was given to a congressman, and the congressman tabled these documents, this data in the Congress, but didn't get a lot of media attention. So anyway, there was a documentary made based on these, and you can have a look at that and make up your own mind, right? So that's a bit of background and some controversy behind Dr. Brian Hooker, which is one of the experts that I used in this video, right? But like I said, I mean, the arguments that he raised here, I feel that they are valid concerns. But anyway, it's up to you to decide. The other academic that I cited is Professor Christopher Shaw. So in his case, actually, it's the I guess he actually did at least some deliberate, some mistake or some deliberate, unethical action, I guess, because there was a So basically, he published this paper on in 2017, quite recently. The about So I mean, it's widely known that vaccines can, particularly vaccines given to children, contain aluminum, right? So this is a talk by Professor Paul Offit. He's actually uh, he's the director of Vaccine Education Center in the US. So he's kind of a very senior person, and I mean, it's standard knowledge that they contain vaccines. So the current accepted knowledge is that it doesn't really, I mean, the amounts that are there doesn't really affect the children's health, right? So anyway, Professor Christopher Shaw published this article, and which kind of suggested that with a mouse model again, that the levels of vaccine adjuvant, and that is the aluminum in vaccine as adjuvant, caused the brains uh, of the mice to show biomarkers of autism. Right? So this generated a lot of controversy, and apparently so it was published, but after publishing, I mean, there are some, I guess they found some errors in the data or results. So Professor Shaw says that they don't, those anomalies don't really affect the conclusion, but anyway, he had to retract it, right? In the previous case with the Brian Hooker's paper, actually, it was uh, uh, it was not him that retracted the paper; it's the journal editor. So sometimes the editor can also retract the journal. In a way, this shows that even if a peer-reviewed journal gets published, article gets published, it can get so it goes to the peer-review process, but it gets published, but still it can get retracted for various reasons. So because of that, of that retraction, since the author himself had to retract, the credibility kind of him was questioned there. But anyway, he is still there at UBC, so I think he's a tenured professor. Maybe that's why it's very difficult to fire a tenured professor, so that's why he's surviving after this controversy, and this was reported in some of the media at that time. So I just wanted to give that background as well, because both Dr. Brian Hooker and Professor Christopher Shaw have been involved, have been questioning about the safety of not this vaccine, some other vaccines. So I mean, maybe whether, I mean, someone would question whether they would have a bias against vaccines in general. Right? But like I said, the questions that they raise in this context, I feel they are valid questions, and at least we should look into those questions. And it's very, I guess it's relatively easy to test these claims, whether there's a DNA or alteration happening from the Oxford-Astrazeneca vaccine. I guess with the available technology, it should be fairly easy to test. The other thing is whether the in mRNA vaccines, whether the mRNA goes into other organs, right? Again, using animal model, I guess should be fairly straightforward to test. I feel, I don't know whether there's any other technical difficulty in doing that. I feel it should be fairly easy to test. Right? So that's basically what I want to talk about in this video. So regarding the key question, whether Oxford-Astrazeneca vaccine, which is a recombinant DNA vaccine, which is the first of its kind to get approved in the US and UK, or in mass production, whether that has whether that can alter the human DNA of the recipients. So my feeling, I mean, after I was exposed to this material, I feel that there is a possibility. That's what I feel, and but I think it can be tested easily and validated whether it really does that or not, right? So I suppose if that research has been done, then I think that should be published, and if it's published, and if I haven't come across that, then maybe, so that's my bad. So maybe someone can put that research in the comment section. And if it hasn't been tested, I think it should be done because I think it's an important problem. So that's it. Thanks for connecting, and most probably this will be the last video that I'll do regarding the vaccines, because it's anyway a controversial topic, but I think it is relevant as well, in these times. So thank you for connecting, and see you again with a similar video. Thank you.